All right, let's dive right in. Today, we're stepping into the world of a high-stakes medical investigation. We're talking about scientists acting as genomic detectives on a mission to solve one of the most puzzling cases in liver cancer.
And this is the core mystery. You can have two patients, both with multiple tumors in their liver, but their futures can look incredibly different. So the question that's been nagging researchers for years is simply, why? What's the secret that makes one case so much more aggressive, so much deadlier than another?
So, here's our game plan, our investigative roadmap. We're going to start with a tale of two very different tumors. Then, we'll crack their genetic code. After that, we'll go inside the tumor's neighborhood to see what's going on. And we'll find an immune system under siege. And finally, we'll connect all the dots to see why it matters and what the future of this fight looks like.
Okay, first things first, we have to understand that not all liver cancers with multiple tumors are the same. Not even close. In this case, we're really dealing with two totally different types of culprits.
On one side, you've got what's called intrahepatic metastasis, or IM for short. The best way to think about this is as a single copycat criminal. One original tumor starts, and then it spreads copies of itself throughout the liver. Now, on the other side, you have multicentric occurrence, or MO. This is way different. It's more like a gang of individuals. Each tumor is a brand new, independent cancer that just happens to pop up in the same liver. They're not related at all.
So, how in the world do you tell these two apart? Well, our genomic detectives needed a way to trace their origins. And to do that, they brought in some seriously powerful forensic tools.
Their main tool is something called whole exome sequencing, or WES. The easiest way to get your head around it is to think of it as genetic fingerprinting, but for cancer. It lets scientists read the tumor's DNA, check for matches, and figure out exactly where it came from.
And when the results came back, they were crystal clear. The IM tumors all had the same genetic fingerprints. They were clones, no doubt about it. The MO tumors, though, all different fingerprints, proving they were all originals. But the detectives found something else, something really important. The IM tumors were way more complex. They had more variety in their genes, they acted more like aggressive stem cells, and their metabolism was just supercharged. Right away, the genetic evidence started pointing a big finger at IM as the more dangerous culprit.
But here's the thing. Knowing the criminal's identity is only half the battle. To really understand what makes IM so deadly, our detectives knew they had to investigate the crime scene itself, the immediate environment around the tumor.
Scientists have a name for this, the tumor microenvironment, or TME. You should just think of it as the neighborhood the tumor lives in. And it's a huge deal because it's not just about the cancer cells. It's about this whole ecosystem of immune cells, blood vessels, and other tissues that the tumor has to interact with. It either fights them or it corrupts them to help it grow.
Now, to figure out who's who in this neighborhood, the team used another incredible tool called single-cell RNA sequencing. This is basically a cellular census. It lets you go in and identify and count every single cell type in that microenvironment. It's like getting a perfect, high-resolution snapshot of the entire community.
And when the results of that census came in, it was pretty shocking. What it revealed was an immune system in full retreat, a system completely under siege, but only in one of the neighborhoods.
Just look at this. The census found a huge difference in these really important immune cells called CD20 plus B cells. In the IM neighborhood, they were almost completely gone. In fact, the MO neighborhoods had more than 16 times more of these good guy cells. It's like a town where all the police have just up and vanished.
And the story didn't end there. Another key immune cell, the CDC2 cell, think of them as the first responders who sound the alarm. They were also seriously depleted in the IM tumors. The body's emergency response system was basically offline.
So what about the immune cells that were left behind? Well, the T cells are supposed to be the body's elite soldiers, right? The special forces that are meant to kill invaders like cancer. But in the IM neighborhood, the census found these soldiers in a state of what's called terminal exhaustion. They were there, but they were just spent, too worn out to even put up a fight.
So at the end of the day, you have these two completely different scenarios. The IM neighborhood is actively hostile to the body's own defenses. It kicks out the good guys and tires out the ones that remain. The MO neighborhood, on the other hand, is much more helpful. It's a place where the immune system actually has a fighting chance.
And this quote from the study pretty much sums up the final verdict of the whole investigation. IM presents more energetic malignant cells and more repressive TMEs than MO.
Let me translate that. It means you've got a smarter, more energetic criminal operating in a neighborhood that has completely taken down its own defenses.
So, we've identified the culprit, we've analyzed the crime scene, but now we get to the most important question of all. Why does this matter? What does all this mean for actual patients?
And this is where all those clues just click into place. The research confirmed, without a doubt, that having the IM type of cancer is linked to poorer survival. But crucially, the data also showed that patients who had more of those B cells and more of those CDC2s in their tumor's neighborhood, well, they had better survival rates. It makes it so clear. The neighborhood isn't just some side detail. It's a critical factor in predicting a patient's outcome.
But this deep understanding doesn't just solve an old mystery. It gives us a whole new way of thinking about how to fight back. It basically gives doctors a brand new playbook.
And here's what that new playbook could look like. Step 1. Use genetic fingerprinting to know your enemy. Is it IMO or MO? Step 2. Analyze the neighborhood. Take that cellular census to see exactly which immune cells are missing in action. And then step 3. And this is the really game-changing part. Tailor the therapy. Instead of a one-size-fits-all drug, you could create personalized immunotherapies designed to rebuild that patient's specific neighborhood to actually bring those B cells and CDC2s back into the fight.
And that leaves us with this incredible final thought. For decades, we've been focused on attacking the cancer cell directly. But what if the next huge breakthrough isn't just about going after the criminal, but about rebuilding and empowering the entire neighborhood so it can police itself? This research suggests that might just be the future of fighting cancer.